Hello, my name is Emilia Moreno, and I'm going to talk about the sugar-related physiological patterns in water foragers of the honeybee Apis mellifera. Honeybees are central-place foragers. They leave the colony to collect resources from the environment and then return to unload before starting a new trip. Usually, a forager departs the nest carrying a honey load that acts as fuel for the flight back home. Honeybees collect both energetic and non-energetic resources, and they handle them in very different ways. Nectar, which is an energetic resource, is a mix of sugars, mainly sucrose, and water. It's carried inside the crop, an impermeable sac often called the social stomach. Pollen is a complex mix of nutrients such as proteins, lipids, minerals, and vitamins. Pollen foragers don't ingest pollen at the flower. Instead, they transport it externally on a specialized structure on their hind legs called the corbiculae. Another important resource is water. Water is a non-energetic resource also carried inside the crop. Although nectar already contains a large proportion of water, colonies still collect water directly from natural or artificial sources. They use it for nest temperature regulation, larval food preparation, metabolic needs, and for cooling or humidifying the nest, especially in hot or dry conditions. Water foragers face a major risk. Their crop are full of water instead of energy-rich nectar, which means they have no metabolic fuel for their return flight. Now, we know that foraging specialization in honeybees has been widely studied through differences in sucrose sensitivity between nectar and pollen foragers. This was tested through the proboscis extension reflex using the gustatory responsiveness score, which measures how bees respond to increasing concentrations of sucrose. Studies have shown that pollen foragers are less responsive to sucrose when they arrive at the food source, but become more sensitive at the end of the foraging visit, probably because they are tired and hungry. In contrast, nectar foragers show the opposite pattern. They start off very responsive, but their sensitivity decreases after recollection, likely because they are sugar satiated. These changes suggest that bees' foraging motivation and internal state dynamically modulate how sensitive they are to sugars. In this study, we aim to explore how water foragers fit into this picture. They collect a non-energetic resource, but one that is also transported internally, just like nectar. So we compare sucrose responsiveness among pollen, nectar, and water foragers, both at the beginning and at the end of their foraging visits. For this, foragers were trained to visit a feeding station located about 40 meters away from the apiary, near a natural water source. At this site, we place feeders containing a 15% sucrose water solution and crushed bee-collected multifloral pollen. During all experiments, we capture water, pollen, and nectar foragers both upon arrival, immediately after they start collecting, and upon departure, just after they finish. To assess sucrose responsiveness, each bee was gently restrained, and we stimulate their antennae with a series of sucrose water solutions of increasing concentrations with a 2-minute interval between trials. Now let's look at the results. In this graph, you can see sucrose concentration on the x-axis and probability of response on the y-axis. The gray line represents water foragers, the orange line pollen foragers, and the blue line nectar foragers. With circles, we have foragers upon arrival, and with triangles, foragers upon departure. As expected, nectar foragers were more sensitive to sucrose when they arrived at the feeder and became less sensitive when leaving, and pollen foragers were more sensitive after recollection. Interestingly, water foragers respond to higher sucrose concentrations when they arrive than when they depart. We would expect water foragers to be highly responsive to sucrose at the end of the visit, since their crops are filled with water, what makes them hungry. These findings suggest that water collection is modulated by crop distension rather than by the individual's energetic needs. And altogether, this mechanism seems to regulate water foraging according to the colony's collective demands, even when the individual's bee's energy requirements are not directly fulfilled. Thank you very much.